Crop diseases systematically degrade global agricultural yields every harvest cycle. Stopping these localized outbreaks before they spread across an entire field requires early, accurate detection at the level of the individual leaf. Mango cultivation represents a massive sector of the agricultural economy, particularly in regions like India, which produces roughly half of the world's supply. But these high-value groves are highly vulnerable to specific pathogens. When inspecting these trees, human experts look for specific biological markers. For anthracnose, a fungal illness, the visual signature consists of dark, sunken lesions spreading across the leaf surface. Sooty mold presents as a thick, black, powdery fungal coat that physically blocks sunlight, actively inhibiting the plant's photosynthetic process. And bacterial canker manifests as distinct, raised, water-soaked spots that eventually rupture. Relying on specialists to walk thousands of acres to manually identify these subtle variations is slow, prone to human error, and entirely non-scalable for modern agricultural demands. Translating these nuanced biological realities into an automated, scalable system requires bridging the gap between human visual intuition and machine perception. Automating this identification is difficult because raw agricultural imagery is chaotic. A camera captures severe environmental noise, shifting sunlight angles, harsh shadows, and heavy layers of overlapping foliage. When standard convolutional neural networks process this unrefined visual data, they frequently struggle with overfitting, failing to generalize across different environmental conditions. If a model relies entirely on its hidden layers to deduce patterns, it often devolves into a black box that memorizes the background noise of the training data instead of the actual plant pathology. To solve this, researchers are sidestepping pure deep learning feature extraction. Instead, they are utilizing a hybrid approach that explicitly engineers the input data long before it reaches the network. This diagram outlines the complete journey from the initial mango leaf image through segmentation and into the neural network classifier. Two blocks represent the core innovations: explicit texture pattern engineering and feature extraction, and a meta-heuristic swarm optimization algorithm. By explicitly dictating the mathematical rules for feature extraction and then dynamically tuning the network's internal weights, this architecture avoids the structural pitfalls of standard CNNs to achieve high classification accuracy. The computational pipeline begins with background subtraction to isolate the leaf from the noisy agricultural environment. Edge detection algorithms scan for the strongest line of symmetry, isolating the leaf's primary central vein to establish a geometric anchor. The system then maps the secondary veins and converts the boundary coordinates, successfully segmenting the target leaf and its diseased areas from the irrelevant background pixels. Even after segmentation, the raw pixel values remain heavily multidimensional and disorganized for optimal neural network processing. Here, local binary patterns, or LBP, are extracted. The algorithm isolates a 3x3 pixel grid, calculating intensity differences between the center and surrounding neighbors. It applies a threshold. Positives become 1, negatives become 0. This outputs a binary sequence that structures local texture data. However, LBP has strict limitations. It is highly sensitive to visual noise and completely ignores directional image gradients, meaning it misses broader structural outlines. To compensate, the pipeline implements a secondary technique, the local directional pattern, which captures edge responses. LDP deploys Kirsch masks, mathematical filters that evaluate the gradient magnitude of pixels across 8 different spatial orientations to isolate the strongest structural edges, like curves and corners. LBP extracts the raw textural surface, while LDP identifies the robust structural edges. Yet neither metric provides a perfectly deterministic feature set on its own. The solution is the local optimal oriented pattern, or loop. It operates as a nonlinear combination of the LBP and LDP methods. Loop mathematically cancels out the empirical restrictions of the directional patterns and the false binary assignments inherent to the basic binary patterns. The final output of this extraction is a highly discriminative, rotation-invariant mathematical feature vector. It is a clean, highly compressed representation of the disease pathology. This graphic represents the neural network classifier. It receives the engineered feature vector at the input, processes the data through its nodes, and outputs a classification determining whether the leaf is normal or infected. The input vector is now mathematically pristine, but the network's ultimate classification accuracy depends entirely on how optimally its internal parameters, the weights connecting these nodes, are tuned. Tuning these hyperparameters is a computational challenge. Standard gradient descent algorithms frequently get trapped in local optima within complex data landscapes, halting the process prematurely. To bypass this, researchers introduced the Coyote-Gray-Wolf optimization algorithm, or COGWO, a biologically inspired model. Think of the hyperparameter search space as a multidimensional topological map of peaks and valleys, where the lowest valley represents the optimal neural network weights. The algorithm initializes a population of nodes across this map. Each node represents a specific, unique sequence of network hyperparameters. The system applies a social hierarchy modeled after the Canidae family. It evaluates the mathematical fitness of these parameters and assigns the nodes distinct ranks: the leader, the representative, and the obeyer. Obeyer nodes dynamically recalculate their coordinates based on the leader's vector. This hierarchical hunting balances exploring new terrain and exploiting known results. If the swarm gets caught in a localized valley, the collective pull of the leader drags them out toward the true global minimum. This dynamic navigation mathematically forces the neural network to lock onto a set of weights that standard optimization would overlook, pushing its classification capability to its absolute limit. When tested against an independent dataset, the fully tuned hybrid COGWO model demonstrates exactingly high performance. Over multiple training epochs, the system achieved a precise F1 measure of 96.47. To verify the model's internal logic, engineers deploy gradient-weighted class activation mapping, known as GradCam. GradCam generates a strict localization map, illuminating exactly which pixel regions in the final convolutional layer triggered the network's classification decision. The high activation zones in red and yellow map perfectly to the actual anthracnose and canker lesions. This confirms the algorithmic perception successfully aligns with the biological ground truth identified by agronomists. Moving this system out of the laboratory introduces a primary engineering bottleneck. The hybrid model, driven by its custom feature extraction pipeline, requires a heavy computational load. The high-end GPU arrays used to train the network are entirely absent in the target deployment environment. The final software must execute inference on a farmer's smartphone, operating in an off-grid field. Translating this research into a viable field application mandates strict model compression. Engineers will utilize quantization-aware training and structured pruning to strip away redundant network parameters, reducing the memory footprint without sacrificing that 96% accuracy rate. Future research factors are expanding the input parameters to include multimodal data, such as hyperspectral and thermal imagery, allowing the system to operate under extreme lighting variations and motion blur. Bridging the gap between rigorous meta-heuristic mathematics and edge-deployed hardware is the vital final step in turning abstract research into a practical, localized tool for securing global food pipelines.